Today we're going to talk about the liver, the gallbladder, pancreas, and salivary glands as we continue with the digestive system. We want to learn the general and unique structural features of glands associated with the digestive tract, and to learn the origin of these glands and how each gland contributes their function to the digestion and absorption of foodstuff, yet how they contribute to maintain a barrier to the environment. We start out with the liver. We can see there's a classical lobule of the liver right here where you have blood vessels in the outside draining through the sinuses ultimately to the central vein. And we can see that here as well, where blood vessels give blood into the sinuses that ultimately is drained into the central vein of various lobules. So there's a liver. And then we have the pancreas. The pancreas we'll be talking about today, where you have the islet cells, intercon part, and the extracon part of the pancreas. So if we were going to look at the liver, a good liver to look at would be a pig's liver, because they have connective tissue that separates the classical lobule, which has a central vein in the middle, and it has several of these portal radicals or portal areas, you would say, that are composed of the portal vein. So you have some blood from the intestines. You have a hepatic artery, which bring in oxygenated blood into the sinuses, and then you have the bile duct. So three basic things make the triad, and we can see those here in this piece of liver from a pig, and you can see the classical lobule here with connective tissue around it. Blood vessels from these different locations, the triads all around, would ultimately drain into the central vein. And so here we can see the triad. You see the cuboidal cells of the bile duct. You see the endothelial cells and the muscle there of the arteries. There's one and two arteries. So usually just one, but there's two there. And then we have the hepatic vein that also provides blood that percolates through here, and you can see where it percolates down the sinuses ultimately into the central vein. While it's percolating through there, it would be detoxifying things from the small intestines. So if we see a part of the liver lobule, you can see where you have the arteries in through there and the portal vein bringing blood, mixing the blood together, and then ultimately it goes through these sinuses to the central vein. If you look within these, you can see Kupffer cells. Kupffer cells, where here they've been injected with Indian ink, that you can see in this artist's view. Those are located at the same places you have endothelial cells, but they're phagocytic cells from the macromonocyte lineage there. And then here we can see another piece. This shows you the bile canalicus. So you can see the little black line in through there. So it shows you where the bile is running. Actually, the bile runs opposite of the blood and goes here to the bile duct that would be there. Here is another interesting component where you can see the mitochondria. There's lots of mitochondria in the hepatocytes, and they're red here, so you can see them. And also the blue is the fat droplets, so there's fat in there. In addition to that, there's a host of glycogen. So you can see glycogen stored in the hepatocytes. Surrounding each of those hepatocyte columns would be the reticular fibers. So you have reticular fibers which branch to support the cords of these hepatocytes, and then they also support the sinuses throughout and the central vein. So blood enters here, percolates through there as it goes to the hepatocytes, it detoxifies things, and then through the central vein, and from there back to the vein to the heart, and then circulation. So we see a classical lobule again with the, here we can see the bile duct here and here, and we can see the hepatic vein in through there, and then the central vein where blood would be percolating through these sinuses in through here. Again, we can see the central vein, which would be draining the material from the hepatocytes. But mostly what we see there are nuclei of the hepatocytes. If you look at one of those triads, you see the hepatic vein. You can also see portal vein. You see hepatic artery and the bile duct that we see there. So those are the things you can see: portal vein, bile duct, artery. Those are the triad components. And here we can see the Kupffer cells. Some of the Kupffer cells that project across the lumen there would be Kupffer cells. And in this particular slide, 118, you can see where colloidal carbon has been injected into the animal, and as a consequence, we were able to see the carbon uptake by the Kupffer cells. Here we can see a higher mag of that with the Kupffer cells showing you the carbon deposition inside there. So the macrophage cells were eating the carbon material that allows you to visualize them. Here, so we can see the sinuses as well as you can see the hepatocytes. A big nucleus and a nucleolus that we see there, endothelial cells lining these sinuses. If we look at the electron microscopic level, you can see the host of mitochondria that are in the hepatocytes. You can see rough endoplasmic reticulum. You can see glycogen storage, these rosettes of glycogen, autophagic vacuoles. And so these hepatocytes that we see there with a big nucleus and distinct nucleolus would be important in making ribosomes for this endoplasmic reticulum. If you look at the sinuses, you can see the endothelial cells lining the sinus. In fact, the endothelial cell also lined the space of Dease. The space of Dease is the space between the endothelial cells and the hepatocytes, where the fluid actually bathes the hepatocytes in through there. Well, here we see a bile canaliculus, another one right in through there. In fact, we can see a bile canaliculus here in these hepatocytes cords that we see. So these would be the Kupffer cells, endothelial cells that are located in through there, space of Dease, and there's another Kupffer cell or endothelial cell. You can't tell right there because it doesn't appear to be phagocytic. If you leave the liver and go to the gallbladder has these projections in through here, and the epithelium is simple columnar epithelium lining those. And there's smooth muscle associated with discharge of the contents of the gallbladder. So the gallbladder here, again, simple columnar epithelium. You can see spaces between adjacent cells where they kind of opened up a little bit. Nuclei of these cells, lambda propria, you can see plasma cells, nice big plasma cells in through there, simple columnar epithelium with wide spaces at the base of them as they're actually pumping out a salt solution and making a higher concentration of the bile. We can see plasma cells, very nice plasma cells in there producing antibodies that will be discharged into the bile. If you look at this portal vein and bile duct set that we see here in the monkey, here is the portal vein, the large vein that we looked at before. Here's a common bile duct and the cystic duct. And the cystic duct is the one that goes to the gallbladder itself and regulates a discharge from the gallbladder. And here we can see those. This is a common bile duct coming through here, and then this would be the cystic duct right in through there, and the gallbladder. And the common bile duct is the one that's going to discharge in the small intestine. It discharges its contents whenever there's a fatty meal to emulsify the fats inside there. So that's the liver and the gallbladder. Then we have the salivary glands. Salivary glands, different ones located in the oral cavity or right off of the oral cavity. And interesting thing about the salivary glands is they have these striated duct. They're striated ducts that we see little lines inside there, and we'll learn in a minute what those are. There's also an intercalated duct, the first little duct that comes off of the secretion. And sometimes the secretion is mucus, and sometimes the secretion is serous type cells. But in either case, there's myoepithelial cells. You can see where they have wrapped around these myoepithelial cells that contracts to discharge the contents within the lumen. So here we see a striated duct, one of these ducts in through here, and there's striations because you have infolding of the plasma membrane that you see here, and that lines up the mitochondria. So actually what we're seeing there is lined up mitochondria that gives its striation. We have the intercalated duct, the very first duct. After the secretions, you have the mucus, which has a flattened nuclei and a flocculent type of secretion there, and then you have a serous type cell that produce more proteinaceous, more carbohydrate, more proteinaceous component, and the striated duct. So here we can see a salivary gland showing you the glandular secretion portion and the striated duct. There's a striated duct right here, here and here. This is the serous and the mucus secreting parts of it. Again, mucus, serous secretions. The serous secretions have a more spherical nucleus as opposed to a flattened nucleus at the base. And here we see the striated duct. There's the intercalated duct right in through there. There's the intercalated duct that runs right in through there. And as I mentioned, the striated duct is due to the infolding of the plasma membrane and the lining up of the mitochondria. So you can see these striations in through very nicely, which are lined up mitochondria. So there are different portions of the duct. There's a secretory portion, the intercalated duct, the striated duct, and the excretory duct. The excretory duct usually starts out cuboidal, and then it becomes columnar, and then stratified columnar as it increases in lumen size and the size of a duct. So in the secretory portion, you have lots of rough endoplasmic reticulum associated with the secretions. In the intercalated duct, you'll have some, but really they're not contributing to secretions that much. And in the striated duct, you have lots of mitochondria located at the base because they're pumping out water. So they are concentrating the fluid. What happens is the secretions here is isotonic, about 300 milliosmoles. However, in order for you to mix saliva with foodstuff, it'd be better if it was hypotonic. So you're pumping out the electrolytes. That's what these increased plasma membrane area here, which is just kind of the opposite of microvilli on the surface by having infoldings inside the cell, and lots of mitochondria for those secretions to occur. So we have the excretory duct coming out. Again, it starts the hierarchy of ducts starts with a simple columnar, simple cuboidal, and then stratified cuboidal cells. So here we see a duct. We have a serous and the mucus type secretions, and if you have those two together, we call that a demilume. A demilume. So this is a serous demilume right in through there. In either case, you would have myoepithelium surrounding the cells. For 40, we will see the glandular secretion portion, but also we see the excretory duct. This is the excretory duct here, which has, this is stratified columnar, as I mentioned to you before. Connective tissue all through there. Here's a nice acinus here with a mucus type secretion with flattened nuclei. Here again we can see another salivary gland. These are the serous type, which the cells are more spherical in shape and maybe the nucleus is not at the base, and then we see the striated ducts. You can see striations very nice here, which are the mitochondria lined up. As I mentioned to you again, you can see the many striations that we have there. So in addition to the liver and the salivary glands, we have the pancreas. The pancreas has both an endocrine function and an exocrine function. Exocrine function is the secretions, pancreatic juices that help in digestion to occur. And we see those pancreatic acinar cells that we've studied from the very first day. This is an acinus. This is a little intercalated duct in through there. So these are the pancreatic acinar cells that Pilati used to study secretions to occur. So in addition to have the pancreatic acinar cells, we have the islets of Langerhans. These are the pancreatic islets which produce insulin and glucagon. And the reason they're distributed throughout the pancreas is there is a portal system between these endocrine cells and exocrine cells. And so by having them distributed throughout the pancreas, you have an opportunity for a local portal system to work. It's actually an arterial portal system that runs from the islets to these other cells. Insulin stimulates them, glucagon inhibits their secretion. But we see an acinus right in through there. In the center, we see the central acinar cells, which actually is the starting of a duct. And here we see an intercalated duct. So this is the endocrine portion. We have beta cells, alpha and beta cells. And then here we also have the pancreatic acinar cells. Pancreatic acinar cells make an acinus, as you can see, with the central acinar cells in through there and the endocrine component. Here in this slide 36.7.23, we can see capillaries in through there. There's an intercalated duct right in through there, and you can see the kind of grayish stuff is the actual secretions of the pancreatic juices. We can also see a nice big cell, pancreatic acinar cell with a big nucleolus there, and the granules in the apical region to be discharged into this lumen here, which appears to be a gray. So this is where you get concentration of the granules and then the condensation of the contents therein, so that it discharged into the lumen. So here we see the islets of Langerhans, which have the islet cells, beta cells, and alpha cells that are located there. Here again, we can see intercalated ducts right in through there. The secretions from the pancreatic acinar cells. This is a larger ducts that are coming in as cuboidal cells located around them, and then we see one of those islets Langerhans, and we can see some blood vessels in through there, which would be part of the portal system that goes from here. First capillary network here, second capillary network is over in that region. Again, an intercalated duct, very nicely one right here. As a longitudinal view of one, you can see the gray stuff inside there, which is the secretions, pancreatic acinar cells, a little blood vessel right in through there. If we look at this electron microscopic level that we looked at the first week or so, we can see a big nucleus, big nucleolus, lots of rough endoplasmic reticulum, zymogen granules as they be discharged into the lumen. So these granules here are the same as these. This smooth area right here is where you have the rough endoplasmic reticulum. So what we've talked about today is the liver. Here's the liver. We talked about the salivary glands. We also talked about the pancreas, and the gallbladder is what we talked about today. So the liver, gallbladder, salivary glands, and pancreas is what we saw today. So in terms of the whole GI tract, we start in the oral cavity, which has stratified squamous epithelium, and go through the esophagus. Again, we have stratified squamous epithelium with the different layers, muscularis externa, as well as muscularis mucosa. And then we have the stomach. We have the surface mucus cells. We have the parietal cells, chief cells, And then we get to the small intestines. We have the Paneth cells, the Argentophan cells, intestinal absorptive cells, goblet cells, and then in the large intestine, many goblet cells, no villi, just we have the crypts, muscularis mucosa, and a lot of lymphoidal components. And then today we talked about the liver, gallbladder, salivary glands, and the pancreas, the striated ducts that you saw in the salivary glands. You do not have striated ducts in the pancreas. It's in the salivary gland to make the saliva hypotonic.